The Human Genome Project by Caleb Verbrugge. What is it? The Human Genome Project is the main project conducted by a whole bunch of biological geniuses to sequence the original human genome. The original genome that was sequenced was from 2 or 3 donors that wished to remain anonymous. This took 13 years to get most of their DNA sequenced. There are some gaps, but for the time, 80% of the genome was a huge breakthrough.
Why is this so amazing? I mean, just having a genome with a lot of letters doesn't really matter much. Well, having the human genome of a person means that scientists can interpret the Hg's, T's, and C's to find the chances of a person getting any type of genetic disease that we know of at the moment and know which pairs cause them. We used to know only about how many pairs we had, but had no idea what they did. But now we know that there are a few different spots that can indicate if someone will get cerebral palsy, or a few types of cancer, or any number of genetic diseases. Having a genome can let the person with the disease know that they need to take preventative medicines, possibly even genetic preventative medicines. It's also amazing because we will eventually know what most of the H's, T's, and C's of the DNA of a person does, and possibly even know how to edit them in a beneficial ways. This has unlimited possibilities. We could have glowing hair, for instance. That'd be really cool. The glowing gene in a firefly's tail has already been inserted into cats, so they have glowing fur or skin. This cat is lying next to a normal cat. That is really creepy. The basics of sequencing. There are 4 major steps that will be explained in more detail later. Step 1. DNA sample preparation. You break the DNA into pieces of 400 to 600 pairs, attach adapters, and separate the DNA into single strands, so half of the basic double helix. Step 2. You add each of the fragments with adapters into containers containing synthetic oil. You shake the container, and that causes the water and oil effect, so like little bubbles of water containing the DNA and the adapters in the middle of the oil. You isolate them and then duplicate about 10 million times. Step 3. You load all of the capture beads, which are the fragments with the water. You load the enzyme beads and into the plate, and then you put it into the sequencer. The plate has tons of little holes about the size of half or about a third of the tip of a hair. It's really, really small. Step 4. You press the start button on the sequencer. That floods the plate with reagents for HGCs and Cs. They're pulsed and reactions monitored. So like at one point, all the reagents for A's flood the plate. And then if it bonds with one of the strands of an A, it emits a light. The lights are then recorded, and then you pulse all of those a ton of times until there is no pulsing left, and then you take all the recordings and then translate that into a genome. Step 1 of sequencing. You break the double helix of a fragment of DNA. You attach adapters to the DNA fragments. You separate the fragments and adapters into single strands. This step can typically be done by one person in an afternoon without any equipment. Step 2. You add each of the fragments with adapters to containers containing synthetic oil. You shake the container, causes the water and oil effect, and small bubbles of DNA and water are in the middle of the oil. You isolate those by draining out all the oil, and then duplicate about 10 million times. Each duplication is called a capture bead. Step 3. You'll load the capture beads into a plate with a ton of little holes that are about a third of a human hair's width apart. Then you load enzyme beads, which react to A's, G's, T's, and C's. Then you put it into the sequencer. Step 4. You press the start button on the sequencer. That floods the plate with reagents for A's, G's, T's, and C's. Those are all pulsed and reactions are monitored. The reaction is just an emission of light, and when those lights are recorded, then they can later be translated into the A's, G's, T's, and C's. Each A, G, T, and C are pulsed many, many times until no reactions are coming anymore. Then the sequencer translate those onto H, G's, T's, and C's. Kind of step 5. Then scientists cross-reference genome with a regular genome, as regular as a genome can really get, because everyone has something that's different than the majority of people. Then they find differences and attempt to interpret those differences. The future of human genomes. They hope to make it a lot cheaper. It currently costs around $5,000 to get your genome sequenced, and that's at the cheapest. $5,000 is more than a lot of people can swing, and everyone is trying to get the price lower. The process currently takes around 12 weeks, but it's constantly decreasing. It originally took 13 years for one about half-done genome that was very inaccurate, so they're trying to get it a lot faster. They're also trying to get it more accurate. Here's a quote from one of the owners of Complete Genomics, who's actually not very big in the industry anymore. "We have higher quality, but in the research market that doesn't matter very much. Researchers can work with lower quality data. It's really been a startling revelation to us that despite the community saying quality is everything, quality really isn't everything." Quality isn't everything? In this specific of a thing, how is that okay with people? Well, it's not anymore. And now that people realize this, companies really need to work on being more accurate. And in the future, they will be more accurate. Why we need more genomes. We need to have people with known diseases get genetically tested so that we can compare their genome to those of people without the disease and see what's the difference, so we know what we can change and possibly edit in the future with medicine. We also need to know if there are easy ways to counter diseases like cancer with genetic medicine, but we still need to know what causes those diseases. How genomes can help. Some diseases, like cancer, have ways that someone can decrease their chance of getting them. Doctors can also give someone preventative medicine, but if someone doesn't know that they have double the chance of a normal person to get the disease, they wouldn't know that they need the meds. Genomes can signify the chances of someone getting some disease, some of which can be life-threatening. Costs. Companies like Illumina are bringing the price of genome sequencing down a lot. They currently have a $5,000 genome, and compared to the original $5.7 billion, it's including inflation, that's amazing. Illumina has also announced that they are in the process of making a $1,000 sequencer. Some people say this is hypothetical, but I read the announcement article, and it sounded like they're almost there. This means that any person will be able to get tested, not just the ones with deeper wallets. This is amazing, and it will save millions of lives in the future. The top companies in genome sequencing. Illumina currently has a $5,000 cost per genome, but is trying to get that to under $1,000. This is just the cost of materials, not the labor and energy it takes. Life Technologies is also very close to the $1,000 genome mark. Oxford Nanopores also has a sequencer plan that costs less than $1,000. Complete Genomics currently has a sequencer at about $7,600 per genome. The three with the plans for the $1,000 genome are way ahead of the other ones, but I mentioned Complete Genomics because they currently have the most accurate sequencer in the market. How many diseases per chromosome? Chromosome 11 alone affects about 93 diseases and or syndromes, including 2 different types of cancer. This is only one chromosome, and it is known to cause 2 different types of cancer. Getting your genome sequence can let you know if you will get or will not get these diseases and syndromes. Why should we care in West Michigan? The University of Michigan has a sequencing branch. They mostly just do fragments of DNA, but they can do genotyping. This can save anyone's life. If someone somehow has a genetic disease that's fatal, If that person got their genome sequenced, they would know and be able either to prevent it or at least slow it down. Using your genome can also tell the sequencers what pairs affect what, so it could save thousands of people's lives in the future.